This video explains the basic parts, layouts, and interface of fast protein liquid chromatography, or FPLC. It describes the setup of the machine and basic operating procedures. Your basic FPLC has buffers, fraction collectors, program interface, pumps, several valves, and different meters. This is a reference image that you can use in the future for the course of the proper labeling of those components. The flow diagram of an FPLC has buffers A and B usually. That goes through a mixer, to a pump, to an injection port, to a column where the separation of protein would actually occur, to several different meters that collect data, and then to fraction collection and waste collection. The FPLC system itself also has a computer that records data from the UV and conductivity sensors that are in line with the flow of the FPLC. The FPLC control panel has several options which we'll discuss and how to use later. As you can see here, this is the control interface. This is the pump that drives the flow of buffers through the system, which operates via a peristaltic mode of action. You can take it apart to load the tubing inside. The inlet to the pump comes from a mixing device that mixes buffers A and B. You have the two meters, the column, that's the UV detector, and the conductivity detector. And we have the fraction collector that collects samples after separation on the column. Now let's take a closer look at the flow pattern through the FPLC. The buffers, in this case buffer A and buffer B, which are used for the affinity chromatography column that we have set up on this device, have two sample lines. They flow to a mixing valve that has two inlets, one for buffer A, one for buffer B, that mixes the lines and have one port that comes out. The mixer can be set to program at different amounts of buffer A and buffer B. The line from the mixer then goes to the peristaltic pump to drive flow of the buffer into the rest of the system. This is the inlet to the peristaltic pump. The peristaltic pump has a special set of tubing on the inside that has two spacers to keep it from moving and interface with the rest of the system and actually drive the flow. This is how you load the pump. Here's an additional view with better view of the insides of the pump, where you can see the rollers and the peristaltic pump and how the tubing fits in the pump. Flow from the pump leads to the switching valve. The switching valve allows for switching between loading your sample and injecting your sample. Sample is loaded into the sample loop that goes from the left to the right via the syringe, and it has a line that goes to the waste and a line that goes to the column. This is the waste line. This is the sample loop, which comes in various sizes. In the load setting, sample is injected via the syringe into the sample loop and then into the waste. This allows you to load your protein sample into the sample loop, and the pump goes directly to the column and not through the sample loop. In the injection setting, flow does not go from the syringe into the system. The syringe goes directly to waste, but the flow from the pump goes through the sample loop, through the switching valve, and then into the column, and the rest of the system allows injection of your sample. Here you see the line leaving the switching valve to the column where the separation of protein would actually take place. Different types of columns are available for the FPLC system depending on the type of protein separation you desire. This is an affinity chromatography column for purification of his-tagged proteins. The line from the column then goes to the UV detector, which detects different levels of protein in the line. And flow from the UV detector then leads to the conductivity detector, which detects changes in buffer composition. These two detectors output to the computer so that you can record results to generate a chromatogram from your FPLC run. Flow from the conductivity meter goes to the fraction collector, which allows you to collect fractions that have been of the protein that have been separated from the column. The fraction collector itself has a control interface, which interfaces with the rest of the FPLC and also has a manual and advanced mode. It has a rack that holds tubes that move and allow for protein to the liquid going through the system to collect in the fraction collection tubes. To inject a sample into the FPLC, you load in a syringe and then attach the syringe via the lure lock fitting on the switching valve. When you have the valve in the load position, it allows you to inject sample from the syringe into the sampling loop. The sample loops come in various sizes and run from the left to the right on the FPLC valves here. And when in the load position, it goes through the sampling loop and then into the waste line. Once your sample is loaded in the sample loop, you switch to the inject mode. In the inject setting on the switching valve, buffer from the pump goes into the switching valve through the sampling loop from the left to the right. During this process, it flushes the sample out of the sampling loop through the switching valve and through the line to the column for the separation of the protein to actually take place. The FPLC has an interface which has three modes. It has a manual mode, a program mode, and a run mode. In the manual mode, there are several options, including the start of flow, purging of the samples, a flow control setting, and a selection of the buffer you wish to flow. In the program mode, this allows for steps to be input to program the FPLC for the exact settings that you want. It allows you to select your pump, the fraction collections, the flow rates, and set up a program that you can actually run. It also has an alarm mode for certain settings. The run mode actually runs this program. This has been an explanation of the fast protein liquid chromatography, or FPLC, basic parts, layouts, and interface.